Hello and welcome to this video. In this video, I'm going to be going over A-level biology, biological molecules, and sort of going over the exam practice walkthrough. I'm going to be going over exam questions and helping you, think about what your answer should be alongside the mark scheme as well, and also explaining through some of the main topic points. So, starting off with the first question, what is a monomer? So, you can see this right here is a monomer. Okay, and this whole molecule right here is a polymer. So I'm just going to write it next to it. So monomers, essentially what happens is monomers join up and they make chemical bonds with other monomers to make sort of long chains, and these are called polymers because they are made up of many molecules, many monomers. And I will just put up the specification for A-level biology. I have opened up the monomers and polymers section, and you can see here the definition. Monomers are smaller units from which larger molecules are made. I'm going to write that as my answer. Okay, monomers are smaller units from which larger molecules called polymers are made. And let's have a look at the mark scheme. The mark scheme says a monomer is a smaller unit from which larger molecules or polymers are made. So that would give us that full mark in that. Another thing about monomers is that monomers and polymerization, which is the process by which polymers are made, is that this process eliminates water. So, for example, when you have two monomers forming to another, one monomer making a chemical bond with another monomer, this eliminates water in that reaction, in that process. H2O, a molecule of H2O, is eliminated from that reaction. So it gives us a molecule of water. On the other hand, when you want to break that bond, that chemical bond, say between two monomers, then you have to put in water to break that chemical bond. So that takes in water. So the breaking of that chemical bond is called hydrolysis. Okay, and the making of the chemical bond is called condensation. So the way that I would remember it is hydrolysis. So hydro stands for water and lysis stands for splitting. So it's water splitting. You are taking in water to split this chemical bond, to break this chemical bond. And when making the bond, when making these two polymers join together and make that bond between them, you are that reaction is essentially producing water. And if you think about condensation on a window and things like that. What happens? Water is formed from that process. So I would think of it the same way. Condensation is when water leaves that sort of reaction. So water is produced as part of that condensation reaction. So that is a way that you can easily remember these two. Right, let's move on to this bit. So I wanted to give you an example of this happening. These are two alpha glucose molecules. Alpha glucose and alpha glucose. So what happens is that these two alpha glucose molecules can join together they are two monomers that can join together. And what happens in this reaction, this is just an example, this hydroxyl group, which is this one here, and this hydrogen from this other hydroxyl group on the other molecule, they join together, and then this reaction leads to H2O being produced. So when these two bond together, H2O is eliminated from the reaction. Okay. Right, so moving on to the next question that I have here. Name the monomers from which a maltose molecule is made. So for this question, we have to pay attention to this part here, and let's revise on what maltose is. So maltose is a disaccharide, and what happens in carbohydrates is that you have got a monomer, and this would be called a monosaccharide. This one, which is when two of these monosaccharides join together, they form disaccharides. And when many join together, this chain could be even longer. These form polysaccharides. So in this question, they are asking about the maltose, which is a disaccharide. So let's have a look at what these form. So maltose is made up of glucose and another glucose, which gives maltose. Okay, and that is the answer to our question. So you can say 2 glucose molecules. Right. To revise the other ones, let's go over them. So for the other one, other disaccharides, you've got glucose plus fructose, which gives sucrose. This is a type of this type of disaccharide, sucrose, is present in fruits. Then you've got glucose and galactose, and this gives lactose disaccharide, which is present in milk. Right, so moving on to the next question. Name the type of chemical bond that joins the two monomers to form maltose. This bond is called a glycosidic bond. And when this sort of, and this is with all the monosaccharides, what happens is the two molecules, when they join together, this side, so this is what it kind of looks like. Okay, this forms a bond like so, and this is called a glycosidic bond. So these two, what happens is this O and H form H2O, and then this oxygen is left, and that forms a glycosidic bond. Okay, I'm just going to write that down here. Oh. Glycosidic. Okay, that is really bad. Let me write that again. Bond, like so. Right? And let's check out what the mark scheme says. So this says glucose and glucose. You could have just said glucose and that would have given you the mark. What I do suggest is two glucose molecules joined together. That is what I've written. Name the monomers. So you can just say glucose because they both are glucose anyways. And then the chemical bond is glycosidic bond. And I will just run you through quickly what this means. Alpha 1 to 4. So this 1,4, which is essentially what it means is that it talks about the carbon bonds that they have. So in here, this is essentially maltose being formed because it is glucose and glucose. So in this reaction, this example is basically about maltose. What essentially happens is that these you have to know that these are carbons, and these actually have got numbers attached to them. So let's have a look at what numbers these have. So this one, for example, would be carbon 1, right? This one would be carbon 2. This one would be carbon 3. This one would be carbon 4. And this one would be 5, and the top one would be 6. So there are 6 carbons in glucose. If you remember the chemical formula for it, it was C6H12O6. So that is what you have here. You've got 6 carbons on that glucose. And then let's do the same thing for this one. 1, 2, 3, 4, 5, and 6. So what is happening here? Between which carbons is the glycosidic bond being formed? It's between 1 and 4. So that is why we call it 1 to 4 linkage. And that is what they have written here. Where is it? That is what they have written here. Right, moving on to the next question. So you've got this one here. Raffinose is a trisaccharide. So this means 3 monosaccharides joined together. Of 3 monosaccharides, so they've just said that in the question, galactose, glucose, and fructose. These are monomers. These are monosaccharides. The chemical formulae of these 3 monosaccharides are like so. Give the number of carbon atoms, hydrogen atoms, and oxygen atoms in a molecule of raffinose. So essentially what is happening here is that there are 3 molecules like so. So you can tell that from these reactions for the condensation reactions to occur, to form these bonds, you have had to have water come out each time. Okay, water comes out each time these form. So there are 2 hydrogen, not 2 hydrogen, there are 2 water molecules that have left as this trisaccharide formed. So that is what we have to take into account, that 2 molecules of water have been eliminated in the bond making process in this big trisaccharide molecule. So what we have to do is add these together. Okay, so I'm just going to add these right here. Let me do that a bit better. So my final one would be C18 and H, what would it be? 36 and O18. But then we have to minus the 2 H2Os, okay, which would be 4 hydrogens and 2 oxygens leaving. So all we have to do, my final molecule I know would be C18, the same because there's no carbons leaving. There would be 4 hydrogens leaving, so we have to minus 4 from this, which my answer therefore would be 32. Therefore, and then you've got 2 oxygens leaving, so my final answer would be 16. Okay, and then they've told you to sort of categorize these here. So how many carbon atoms? 18. How many hydrogen atoms? 32. Oops. And then 16 here. So let's see if the answer is right. Okay, and it is 18, 32, and 16, and no other answers are accepted. So you have to make sure that your calculation is correct, and this is a 1 marker question. So you can actually, what I do recommend is that if you're doing questions like this, definitely sketch it out so you can visualize it in your head. What is happening each time this monomer binds to another monomer? What is my final molecule going to look like? How many hydrogens are leaving this sort of structure? How many, not hydrogens, how many water molecules are leaving being eliminated as a result? And then you take that into account in your final answer, and that should help you figure it out. Right, let's move on to this last question here. So starch and cellulose are two important plant polysaccharides. The following diagram shows part of the starch molecule and part of the cellulose molecule. You've got starch, which is shown here, and you can see the glycosidic bond. So this right here is a glycosidic bond. This right here is a glycosidic bond. And then you've got cellulose. So this right here is a glycosidic bond, and this one right here is a glycosidic bond. These oxygens, these ones right here, these are part of the glucose molecules. Starch and cellulose are polymers of glucose. But what is the difference between, why the structure is so different? Let's revise that. Right, so here you've got alpha glucose, and here you've got let me write that properly, beta glucose. Okay, and you would write these as alpha glucose, like so. This would be the symbol for alpha glucose, and this would be the symbol for beta glucose. Okay, so essentially these are the same molecule, but they are arranged differently. So what I mean by this is that these are both glucose, so they're both C6H12O6, right? So they have same number of carbon, same number of hydrogen, same number of oxygen. It's just that the molecular structure is slightly different. So these are essentially called isomers. So these are isomers of glucose, right? So if you look at it here, let's look at just alpha glucose. Okay, so if you look at this and this, you can see that both H's are at the top and both OH's are hydroxyl groups are at the bottom. Whereas if you look on here, you can see that they are different. So this side is flipped. So let me just take out this blue. Okay, so here these two are flipped, inverted for the beta glucose. And that is, you should remember the structure and how these are different. So you should remember how these are. I do recommend drawing it so that it will help you remember. So what happens is that these two form different polymers. This one will form a polymer, and what do you call that for carbohydrates? You call it a polysaccharide. Okay, so you can call this a polysaccharide. It's just mean carbohydrate polymer, polysaccharide, starch. So that polysaccharide, that alpha glucose will form. So alpha glucose will form as starch, and the polysaccharide that beta glucose will form is cellulose. Okay, cellulose, which is, if you remember, or if you don't, it's fine. Cellulose is part of the makes up the cell wall of the plant cells. So that is what cellulose is made up of, right? And starch is an energy storage molecule that, when it's broken apart, it releases energy for respiration. So explain the difference in the structure of starch molecule and the cellulose molecule shown in the diagram above. So you can just say what you know from this is that starch is made up of, so let me write that here, starch is made up of alpha, what is my pen not writing, glucose, monomers, whereas cellulose is made up of beta glucose monomers, right? And then you can talk about the molecular on your a sort of molecular level, what the structure, how is the structure different. So like I said earlier in the video, you can actually name the carbon linkage that is happening here. So 1, 2, 3, 4, 5 and 6. 1, 2, 3, 4, 5 and 6. So where did I say it was flipped? It was flipped on this carbon, and that carbon is carbon 1. So you can say it here that the hydrogen and hydroxyl groups are inverted, okay, on the beta glucose, okay, monomer. So let's have a look at what the mark scheme says, right? Oh, and on carbon 1. Hydroxyl groups, forgot to say that, on carbon 1. So just make sure you write that down, okay? So let's have a look at the answer. Starch formed from alpha glucose, but cellulose is formed from beta glucose. Always make sure to say but in the question. It helps sort of make a distinction between both things. So when it's like a compare question, explain the difference question, you always have to say this is like this, whereas this is like this. Or you can say something is like this, and then you say however this one is like this. So you always have to make a compare statement in your answer. Starch formed from alpha glucose, but cellulose is formed from beta glucose. It's a good habit to have that sort of comparison statement, and you're using comparison phrases in your answer. Position of hydrogen and hydroxyl groups on carbon atom 1 is inverted. Okay, so you can have a look at this answer, and that is a 2 marker here. So actually I should have said carbon 1 atom. Now that I think about it, that is the only thing missing. So carbon 1 atom, because we have to be specific to the mark scheme. But anyways, that is all for today. I hope that this video was really useful and a sort of walkthrough as to how you should answer questions within the topic of biological molecules. I hope that this was a good revision session. Please do check out other resources that I have on my Etsy shop, very useful learning revision resources. If you'd like to request a specific video, you can do that in the comments below. And for tuition sessions, there is a form that you can sign up for as well.